Good afternoon. Welcome to the neurodegeneration session. My name is Alberto Lleó, the head of the memory unit at Hospital San Pau in Barcelona, Spain, and I will be the chair of this session. Today we have three exciting talks presented by excellent speakers in two data Blitz sessions. Each talk is followed by a 5-minute Q&A session. If you have questions for the presenters, please add it to the question box during the session. We will cover as many as possible after the presentation. The first speaker today is Professor Michael Heneka, who is the director of the Luxembourg Center for System Biomedicine at the University of Luxembourg. The title of this teaching talk is Deregulation of the Central and Peripheral Immune System in Alzheimer's Disease. Michael, whenever you want. Thank you.
Alberto, many thanks for the kind invitation. Let's jump right in. So what is the immune system? When we speak about neurodegenerative disease and Alzheimer's in particular, the most concerned axis of the immune system is the innate immune system. And I will briefly introduce you in explaining why this is. In a pandemic like we live through right now, and in our daily lives, we are exposed to various pathogen challenges from viruses, fungi, and bacteria in this environment we live in. And in our bloodstream, there's a constant hunt for bacteria. In this case, you see a macrophage going after Staphylococcus aureus in order to defend the host. And you'll see that he has to take a lot of clever decisions and avoid interaction with the other cells until he can take it up and degrade it in order to protect us. So the innate immune system is our first line of defense. And these cells obviously don't exist only in the bloodstream. They're also in our body as resident innate immune cells. In the brain, we call them microglia. In the lung, alveolar macrophages, copper cells in the liver, mesangium cells in the kidney, beneath cells in the gut, and of course in the bones, the bone marrow macrophages. They all protect us from infection, but they also contribute to organ structure and function. And regarding microglia, they have been first described by Pio del Rio Ortega in the last century or the 20s. And if you want to know more about this, there's a very nice review by Amanda Sierra published in CLIA some years ago. Now, what is happening in Alzheimer's disease? In a nutshell, we have cleavage of an amyloid precursor protein by beta and gamma secretase in a sequential manner, yielding amyloid beta monomers, most known of 1 to 40 and 1 to 42, which to large parts should leave our brain by excretion into the CSF or into the glial lymphatic pathway. If the remaining oligomers are not being locally degraded, they tend to form oligomer and fibrous structures, which can be visualized in blood systems and also as deposits and so-called neuritic plaques within the brain itself. This deposition of beta amyloid deposits is causing an inflammatory reaction, and you see activated microglia cells within the plaque at the left and GFAP positive astrocytes on the right. And ultimately, the disease is characterized by the occurrence of neurofibrillary tangles within neurons that mark the dysfunction and ultimately the degeneration of these cells. Now, why are immune cells in the brain reacting to the deposition of beta sheet structured amyloids? This shows you a picture with Congo red stained frontal cortex and MHC class 2 positive microglia which surround these deposits. And Congo red actually has been licensed by Paul Bötticher in 1870 as a bacterial stain. Now, what's the common structure and the commonality in general between bacteria and the human brain? And these are again the beta sheet structures. Very elegant work by Matthew Chapman has evidenced already in the 20 years ago in a science paper that bacteria very known to us like Escherichia coli, Staph aureus, Salmonella typhimurium carry beta sheet structured amyloids on their surface, which are looking very similar to the ones which are depositing in our brain. Now, this means that this myeloid cell here has been over evolution taught that the beta sheet structured amyloid is a pathogen surface, and it has been equipped by nature with several receptors commonly denoted as pattern recognition receptors. The most famous family is the toll-like receptor family, but there's scavenger receptors, there's CD36, CD47, and several others. And ligation of these receptors then mount two different reactions. One initiates an innate immune response, which aims to kill the pathogen, the opposing pathogen, and the other receptors induce reaction which are aiming at resolving and removing the pathogen by phagocytic clearance. Now, if microglia cells in the case of Alzheimer's disease sense a beta sheet structured amyloid deposit, they react in a similar way. They undergo massive morphological change. You see that they retract all their processes, which are usually outlaid in a widespread manner to scan the environment and take care about the dendritic trees and synaptic structures. In case of inflammatory activation at an amyloid beta deposit, all these processes are lost. These cells start to release, I'm sorry, this is not, they start to mount an inflammatory reaction by releasing complement factors, cytokines, chemokines, nitric oxide, and free radical oxygen species. At the same time, they stop doing a lot of sensible things. So they reduce their surveillance function, they reduce synaptic scaling and production of neurotrophic factors, including BDNFs. And of course, they're impaired in debris clearance. Now, there are more than amyloid beta in these brains, and there's a number of other danger-associated molecular patterns, including chromogranin A as an example, which has been found to be elevated in Alzheimer's disease, in Lewy body dementia, in frontotemporal dementia, and PD. And chromogranin A is usually kept in neuronal vesicles, but when neurodegeneration sets in, then this is being released, and it's as potent in stimulating microglia immune reaction as bacterial cell wall components, in this case, LPS. Now, another member of the DAM family are S100 proteins, in this case, MRP14, and you can see here by work from Markus Kummer in my laboratory that knocking down MRP14 in an APP-PS1 transgenic mouse model halves the amount of cerebral TNF-alpha interferon gamma production. At the same time, these cells become more active in taking up amyloid beta and degrading it. So suppressing the inflammatory response helps the microglia cell to do their job properly and clear debris, in this case, amyloid beta. Now, the inflammatory reaction can be visualized in the brains of Alzheimer's patients, in this case by a PET ligand, PK1195. On the left side, you see that there is enhancement on the cortex and the limbic system, while in the control case, there's only vascular staining. In general, we have to ask the question, what does inflammation, especially chronic inflammation, mean to the brain? And there is a functional role. And we know that cytokines, including interleukin-1 beta, interferon gamma, TNF-alpha, and nitric oxide, are all able to suppress electrophysiological functions. Importantly, hippocampal long-term potentiation, which is an important factor in information storage and consolidation. There's an interesting synergism with excitotoxicity, and elegant work by Sandy Hewitt and Moses Chow, already published in the mid-90s in Neuron, showed that neither an inflammatory stimulus or a glutamatergic stimulus alone is able to lead to widespread death of hippocampal neurons, but together they do very much so. And there is further work by Harald Neumann's group from the early years, like 2000, where she was able to show that inflammation leads to the loss of synapses, axons, and dendrites. More recently, Sun-Yun Han has identified a mechanism by which microglia engulf and remove the synapses in a complement 3 and complement receptor 3 dependent fashion. And Jonas Neher and Guy Brown have identified a mechanism by which microglia can even take up living neurons by phagocytosis if those do not present do not eat me signals on their surface. Now, when does this all set in? By now, we know that amyloid beta deposition starts way before the appearance of clinical symptoms, and data from the analysis of familiar forms of Alzheimer's disease, but as well as of the sporadic type of Alzheimer's suggests that it can occur as early as 2 to 3 decades prior to the first neurocognitive deficits and short-term memory failure, which means that knowing that beta sheet structured amyloids, once deposited as such a strong danger associated molecular pattern, everything thereafter is taking place in the presence of inflammation. Now, all the pathomechanisms which we will consider thereafter are actually under the presence of inflammation. This comes on a genetic background, and the past decade has experienced the identification of various risk polymorphisms that encode either for proteins that are important for synaptic structures and axons, or for the innate immune system. And CD33, TREM2, BIN1, CR1, and clustering are just some of these members which are important contributors to innate immune signaling and pathogenesis. Next to this genetic background, there are more factors which influence inflammation in the brain. And we know, for example, that midlife obesity in young men, which is a strong risk factor 3 decades later to develop Alzheimer's disease, is a source of constant inflammatory cytokine production. We know that poor oral health, and especially periodontitis, is a risk factor, aging by generating a senescence-associated secretory profile of brain trauma, but also infections or in general systemic inflammation. Reduced physical activity is a further factor that impairs our beneficial immune responses and elevates cytokines in the body. And they all impact, and they all are a risk factor, either alone or in concert, for brain inflammation. Now, it's important to note that when we model these circumstances in mouse models, only a small fraction of the innate and adaptive immune system are being shared between mouse and man. This is elegant work by Suk and colleagues showing that under 3 different conditions, burn, trauma, and endotoxemia, the transcriptome of human or mouse macrophages is really different. And if you look, for example, to the toll-like receptor signaling pathways, these are activated pathways here under the conditions of human or mouse endotoxemia in blue, you'll see that there's really little overlap. So when we start to look at single transduction pathways and alterations of those, we need to be sure that the pathway of concern is conserved between both species. Now, within those, Eric Bodek and colleagues in 2017, in a very nice Nature Neuroscience paper, has identified the human microglia core signature. And you'll see that in the core of this core signature, so to say, there's the inflammasomes and the inflammasome products. So the most abundant inflammasome as a major signaling pathway of innate immune activation is the NALP3 inflammasome in microglia cells, which requires the ligation of a toll-like receptor or a cytokine receptor, inducing the transcriptional activation and generation of proforms of cytokines and auto-inhibited NALP3. The next step is the post-transcriptional activation step, which is characterized by several mechanisms of licensing the buildup of an active NALP inflammasome and its maturation to an AST spec. The AST spec then has the outward-directed caspase-1 activity, which cleaves proforms of interleukin-1 beta and IL-18, endospermin D, the cleavage product of the latter is forming in a pore in the outer membrane, allowing for release of the activated cytokines. Now, this system is active in Alzheimer's disease. You see here a microglia cell containing an acid speck. And of course, the acid speck alone would not have told us how active this system is. And so we started to plot for cleaved caspase 1 activity as a sign of NAB inflammasome activation. Doing this in MCI and AD cases, we found that there's a strong increase, in this case in the frontal cortex of AD cases, in caspase 1 cleavage, so to say, NAB inflammasome activation on the spot, while at the same time age-matched non-demented controls don't show such a signal. Luckily, this was completely mimicked in a mouse model generated by David Borchelt and Joan Jankowski, the APP-1 delta E9 mouse model. And you can see when we knock down NAB3 in this mouse model, we completely prevent caspase cleavage and also the elevation of intracerebral interleukin-1 beta generation. This is an age-dependent process which has been detected first at 8 months and remains active until the end of the life of these animals. Now, in a nutshell, what does NAB inflammasome activation do in APP-1 transgenic mice? It activates microglia and reduces its knockdown, reduces A-beta deposition and spine loss. And importantly, the knockdown of NAB3 in this animal model protected from A-beta-induced LCP suppression and spatial navigation deficits. But it also has a direct effect on the spreading of pathology because it leads to pyroptosis and the release of these acid specks into the extracellular space, and the acid specks cause the oligomerization and act as a seed for deposition of amyloid beta in several murine AD models. And lastly, by generating large amounts of interleukin-1 beta, in response to the exposure of microglia to tau isoforms or A-beta itself, we could show that this NAB inflammasome activity drives tau pathology. Now, what about the peripheral immune system? This is work by Dario Tejera in my laboratory using 2-photon in vivo laser scanning microscopy and exposing mice to a single immune challenge. So we recorded the physiology and the activity of the microglia cells as well as A-beta deposition under control conditions, and then these mice can be repetitively investigated after 2 and 10 days. At 2 different ages, at 5 and 15 months of age. Now, here you see that going back here, you see that at 2 days and 10 days in APP-1 transgenic animals, MX04 positive A-beta plaque deposition is arising very quickly, while in knockout animals, this was not the case. And analyzing this further by the more precise description of these plaques, we found that plaque number and plaque size would be suppressed. There was no such effect of, plaque evolving mechanisms in young animals, but in animals at 15 months of age. Importantly, if we quantify the effects on A-beta levels by ELISA, we found that, sorry, this is not working well. We found that A-beta 42 levels would be significantly reduced in aged APP-1 knockout animals exposed to such an inflammatory stimulus, while the latter would increase very quickly the cerebral amyloid beta 1 to 42 load in response to this LPS challenge. Now, hope this works. This is an analysis that shows us the phagocytic clearance capacity. So we isolated the adult microglia after injection of MX04, which labels amyloid beta under in vivo conditions, and then FACS sorted the isolated microglia for their content of MX04 positive amyloid beta. And you see that in APP-1 transgenic mice, there is a decrease of phagocytosis over time, the immune cells are competent, while in the immune cells are compromised by the inflammatory stimulus. There's less and less clearance of amyloid beta, while in knockout cells, the opposite is the case, and amyloid beta clearance by microglia cell is protected under the very same circumstances. So this means that an inflammatory systemic challenge can drive amyloid beta deposition by compromising microglia capacity to clear amyloid beta away, and this is depending on NAB3 inflammasome activity. Now, we observed that there would be, in the case of systemic inflammation in aged but not in young mice, substantial infiltration of peripheral CD169 positive macrophages. And this data are very much in line with data published by Andoneghi and JCI Insight in 2018, further identifying this subpopulation as CCR2 positive. And importantly, preventing this CCR2 transport into the brain was preventing the occurrence of neurocognitive deficits in the Morris Watermaze experiment and test. So this tells us that this infiltration of peripheral cells, especially in the aged mice, most likely contributes to worsening of a neurocognitive behavior. So can microglia cells be only detrimental? And I believe not. And the reason I believe not is very recent work on TAM receptors, which are receptor tyrosine kinases, which have a role in phagocytosis and immune regulation. There are two known TAM ligands, Gas6 and Protein S, and these TAM receptors are being cleaved from the surface by ADAM10 and ADAM17. There are soluble TAM receptors, and TAM receptors are very important for cytokine STAT-dependent regulation that activate TAM genes. They induce the suppressive cytokine S, which suppresses STAT and NF-kappa beta pathways. And then they're cleaved off, and we use those as a biomarkers in Alzheimer's disease. So the cleavage product here in the CSF was our target for determination. And in two different cohorts, the DZNE cohort Delcode and in a cohort provided by Masha Boada and Augustin Royce from the Fundación ASE, we found absolutely identical data sets. And sorry, it's not moving well. So MIF1 alpha, which is another microglia marker, but more importantly, the two TAM receptors, soluble axon, soluble tyro, are very much increased in tau positive, so-called healthy controls, but also in people who AD, where this type of inflammatory reaction seems to burn out. You see that we have the same data in the Fundación ASE cohort. And interestingly, this analysis and this elevation in patients were positively associated with the protection from atrophy. So here you see the brain regions which are protected, including the entire neocortex and the mesotemporal limbic system. So elevation of S-tyro and S-axis was positively correlated with preserved cortical tissue. And moreover, it was also associated with preservation of cognition. So people with high S-tyro or high S-axis levels were less likely to experience a cognitive decline in the years thereafter as compared to people with low S-axis and S-tyro levels, together suggesting to us that this immune reaction would be neuroprotective. having this neuroprotective immune reaction in patients which are supposed to be healthy but tau positive or just suffering from subjective cognitive decline tells us that this is a very early but indeed protective reaction of the microglia cells, in line with the hypothesis that chronic inflammation at later stages is detrimental to the brain. Now, in summary, I hope I could show you that the innate immune system is activated by danger-associated molecular patterns in the brain. Chronic inflammation may impact on brain function and structure. I showed you the example of LTP. Cerebral immune processes are influenced by genetic background and by our lifestyle. Translational research has to consider species conservation of immune pathways. I think there are a lot of unnecessary experiments in the field which could need to be revisited. The NALM3 inflammasome is a central innate immune pathway. Its activation contributes to A-beta clearance deficits, to the seeding and spreading of A-beta pathology, and to the recurrence of tau pathology. An early TAM receptor shedding suggests a neuroprotective role for microglia at the beginning of the pathological sequence. I've shown you data from my laboratory. Therefore, I want to name the people who were doing all this work, people at my former unit in Bonn, our great collaborators in Bonn, Massachusetts, Worcester, Tübingen, Barcelona, Paris, Lille, Galveston, Münster, and Braunschweig. And with this, thank you for your attention, and I hope you have questions.
Well, thank you, Michael. Really great and clear presentation. We have a few minutes for questions. Well, we get questions from the chat. I have a couple of questions. So, I mean, the issue of biomarkers for inflammatory biomarkers in Alzheimer's disease and other dementias is a hot topic, and there are many studies. But still, the question is, how specific are these biomarkers to AD compared to other neurodegenerative dementias? It seems that there are many biomarkers, but that, I mean, but probably whether is the question is whether reflect common processes or there's some of them are specific for AD that can be measured.
Absolutely. So in terms of, I only can, we know that tau or neurofilament only is partly selective for neurodegenerative disease, and brain trauma and other conditions may also elevate these levels. I only can really speak about the inflammatory markers here, and I would say that most of the innate immune reactions which we consider are very broad and very massive responses, which occur in a variety of diseases. So they are not specific for Alzheimer's. We just have not looked in a cohort of Parkinson's or FTD or LBD cases like 2 or 3 decades ahead of the beginning of the clinical symptoms. We probably would see the same TAM receptor shedding signal. And this is a pure speculation, but I believe that TAM receptor engagement is a very common thing and a common response for an innate immune cell. So it may well occur in other conditions as well. Nevertheless, it was very interesting for us to see that it's connected with protection, and high levels would indicate or characterize people who would show less cognitive decline and less loss of cortical atrophy in the subsequent years. And I think that could either help us to distinguish people at different risk stages already very early in the disease, or it could even offer a sign of therapeutic intervention. We have to find out what exactly TAM receptor ligation at such early phases to the disease process means, and whether on a long term this mechanism itself is mediating neuroprotection, or whether it's just a tombstone.
Thank you. Thank you, Michael. It was a great talk. We have to move now to the data Blitz session. So this talk will be given by Jenny Lange, who is a postdoctoral research fellow at the University College London, and the title is DNA damage drives astrocyte dysfunction in Huntington disease.
So today I want to talk about astrocyte dysfunction in Huntington's disease. Huntington's disease is a devastating neurodegenerative disorder which is caused by a polyglutamine expansion in the Huntington gene. I'm interested in astrocytes because in addition to the many crucial functions they have in the central nervous system, they're intimately involved in the injury and disease response. They've also been shown to be involved in Huntington's disease pathogenesis, where they contribute to neuronal dysfunction and show altered calcium signaling and reduced glutamate uptake, as well as other various phenotypes. Here we use embryonic stem cells and induce pluripotent stem cells with varying CAG length, as well as their matching controls, to generate astrocytes, which we mature for 28 days before commencing experiments. We were interested in DNA damage as the presence of DNA damage has been shown in various Huntington's disease models, but also because more recently genes associated with the DNA mismatch repair complex have been shown to be genetic modifiers of the age of onset of Huntington's disease. We started by looking at DNA double-strand breaks using gamma H2X and 5,3-BP1, and we found a significantly higher presence of these DNA double-strand break foci in our Huntington's disease astrocytes versus their controls. We quantified this on the protein level, where we found significantly higher expression of gamma H2X and 5,3-BP1. We also wanted to look at oxidative DNA damage, and we used an 8-OHDG stain, and again this was significantly upregulated in our Huntington's disease astrocytes versus controls. Cell death often accompanies DNA damage, so we used prapidium iodide to label dead cells, and we found significantly higher cell death in our Huntington's disease astrocyte cultures. We next wanted to look at the DNA damage response. So ATM usually becomes activated in response to DNA double-strand breaks, and indeed we found a significant upregulation in our Huntington's disease astrocytes. ATM in turn can activate phosphorylate ACT, and again we found a significant upregulation in the Huntington's disease astrocytes. Phosphorylated ACT in turn can activate downstream proteins such as p53 and RAD51, which can result in apoptosis or DNA repair, and this is where we see a dysregulation of the downstream proteins in the Huntington's disease astrocytes. I don't have time to go into this now, but please come and see me at my poster to have a chat about this. Thank you for the opportunity to give this talk, and also many thanks to everyone involved in this work and at the lab, and thank you to our collaborators. Thank you.
Thank you, Jenny. Very nice data. We have time for one short question. I have a question. What are the clinical implications of this? Can you identify some specific astrocytic phenotype that is associated to like faster progression or early age of onset? Is there any potential for this to happen?
We actually found that DNA damage is very consistent across Huntington's disease lines with like longer poly-Q length. So astrocytes from patients with adult onset and juvenile onset, they show similar levels of DNA damage. So that seems to be a very consistent phenotype regardless of age of onset. Whereas with some other experiments we found that are not in this talks, like metabolism, we actually found poly-Q dependent changes that may be more related to that earlier age of onset, etc.
Okay, thank you. Thank you, Jenny. Okay, great. Unfortunately, we have to move to the next talk. The next talk is the basic science research talk, and the presenter is Dr. María Llorens-Martin, a tenured scientist of the Spanish Research Council at the Center for Molecular Biology Severo Ochoa in Madrid, Spain, and she's the principal investigator of the Excellence Centers of Cibernet. The title of the talk is Human Adult Neurogenesis and Neurodegenerative Diseases, and we will have 5 minutes for questions at the end. Maria, whenever you want.
Good morning, good afternoon, good evening. Today, I would like to talk about some of the latest results of our group focused on the study of adult hippocampal neurogenesis in human subjects. I would like to start this talk by briefly defining what adult neurogenesis is. Adult neurogenesis encompasses the generation of new functional neurons in the adult brain. Therefore, this process provides an extraordinary reserve of plasticity during lifetime. Adult neurogenesis is a very strictly regulated phenomenon, both in temporal and also spatial terms. It occurs in two main adult neurogenic niches, which are the subventricular zone and the subgranular zone of the hippocampus, on which this talk will be focused. Here you can see a representation of the gross anatomy of the hippocampus, where the neurogenic region, the dental gyrus, is represented in red color. Adult neurogenesis participates in many of the hippocampal related functions, such as learning and memory, and also the regulation of emotions. It has been described that adult neurogenesis is impaired in both neurodegenerative and psychiatric diseases. Here you can see how the whole process of adult neurogenesis looks like in a rodent. This process starts with the asymmetric cell division from neural stem cells that are resident in the dental gyrus. These cells give rise to cells that are also proliferative but acquire progressively neuronal features, these immature neurons that are being progressively differentiated towards fully mature neurons, which are integrated into the trisynaptic circuit of the hippocampus. The classic way in which adult neurogenesis has been studied both in rodents and also in other mammalian species is with the use of specific cell markers. Here you can see this is a very simple scheme in which the sequential expression of these markers can be observed, and you can have an idea that the combination of specific groups of markers identifies specific stages during adult neurogenesis. So by using these markers, which have been extensively validated, several works which approach the controversy of whether adult neurogenesis takes place in the human brain found somehow opposite results. You can see in green color how the consensus in the field is that adult neurogenesis takes place, though these green studies found markers of adult neurogenesis by using not only immunohistochemistry but also other approaches, but some works found absence of the same markers. So we wondered, in the light of these discrepancies between labs, we wondered whether methodological differences could underlie these discrepancies. So as you can see, the methodology to study the human brain is somehow complex, both from the ethical and also the technical points of view. So many of these stages or these procedures can alter our capacity to visualize staining with markers of adult neurogenesis. So some of our initial studies on this topic focused on determining whether fixation could influence or could limit our capacity to visualize markers of adult neurogenesis in the human brain. As a summary of those initial results we obtained, you can see here how we processed the whole hippocampus from several subjects, and what we did was to fix a small fragment of those hippocampi during different periods of time. So the number of cells expected to be found in those fragments would be the same. But as you can see here, with short fixation times, only 12 hours or less, we found an abundant population of immature neurons, which are identified by this specific marker named double cortin. And you can see this beautiful staining of these cells. However, when we fixed the tissue for 12 more hours, this beautiful signal completely disappeared and is replaced by a terrible background which impedes the detection of positive cells. So we have to figure out which was the best combination, the best histological combination in our hands, at least, to visualize again these markers when the samples were appropriately fixed. So as you can see here, by subjecting short fixed samples to a specific, not specific, but a particular histological treatment, we could observe again the presence of those cells which could not be observed in the presence of long fixation times. So after having concluded that fixation was a critical factor not met in all the works studying adult neurogenesis, we slightly modified our protocol to detect not only immature neurons, but also neural stem cells and proliferative cells in the dentate gyrus of the human brain. So together with all the previous evidences that I showed before, our works revealed that adult neurogenesis is a robust phenomenon, at least until the 9th decade of life. In fact, our results also revealed that adult neurogenesis is a dynamic process and it matches the maturation continuum that is being observed in other mammalian species. You can see how double courting, this worldwide used marker for immature neurons, colocalizes with markers that are characteristic of distinct sub-stages of adult neurogenesis, ranging from proliferative cells, very immature neuroblasts, neurons in the period of differentiation, and also cells that have been advanced in their differentiation period. As you can see here, there are striking morphological differences between the very immature neurons and the more differentiated ones, all of them being double courting positive. You can see how these morphologies are exactly the same or are very similar to those described in rodents, with the cells increasing their soma and nucleus size and also occupying positions deeper in the granule cell layer. Our results supported previous evidences, not only in rodents, but also in other mammalian species, demonstrating that adult neurogenesis was reduced with aging. We wondered whether the trophic support that these cells could receive in the dentate gyrus was also affected by ages. And we determined that the neurogenic niche of the humans is composed by the same elements that are present in the rodent neurogenic niche in the dentate gyrus, which is an abundant presence of very thin capillaries, remarkable presence of astrocytes, and also microglial cells. We found slight differences in the composition of this neurogenic niche with age, but I want to share with you what we consider is one of the most striking differences between young and aged human subjects. Here you can see an example of how a microglial cell exhibits a phagocytic pouch, which will be likely using to phagocytose this apoptotic cell in its proximity. We saw that despite no changes in microglial density were observed with age of the subjects, the phagocytic capacity of these cells, which is reflected by the number of phagocytic pouches they show, is reduced with aging. And in parallel to these changes in the morphology of microglial cells, we also observed an increase in the percentage of apoptotic cells that were not being phagocytosed by microglia. So these results revealed that the functioning of the neurogenic niche was impaired during human aging. And we thought that this could have important consequences for the functioning of the whole dentate gyrus, not only during physiological aging, but also during the course of distinct neurodegenerative diseases. This is one of the main interests of our lab, to study to what extent the plasticity or the capacity to generate new neurons in the adult brain is being hindered by the presence of neurodegenerative processes. So we started several years ago by studying one of the most predominant neurodegenerative diseases, which is Alzheimer's disease, which is characterized, as you all may know, by episodic memory impairments, neuronal loss, changes in neuronal cytoarchitecture, and also marked synaptic alterations in distinct brain regions. Moreover, recently we have also studied other neurodegenerative diseases that do not target directly the hippocampus, but are known to have also some secondary symptoms that could be underlaid by hippocampal dysfunction. We studied patients with ALS, Huntington's disease, Parkinson's disease, dementia with Lewy bodies, and also patients with frontotemporal dementia. So we compared two cohorts of subjects, 45 patients with Alzheimer's disease and 38 subjects with distinct neurodegenerative diseases. All of them were compared with a cohort of 14 neurologically healthy control subjects. We did these experiments with the invaluable health of Alberto here at the CM Foundation in Madrid. And it took a while for us to get this important collection of human samples. So our first results was the fact that adult neurogenesis, or at least the number of immature neurons that were detected in the brain of patients with Alzheimer's disease, was remarkably reduced during the advance of the disease. Importantly, we found a 33% reduction in the number of immature neurons in patients at Bractal stage 1, which means that the alterations in adult neurogenesis may be earlier than the appearance of cognitive impairment or neurological symptoms in these patients. This decrease was specific for immature neurons, as the number of total mature neurons was unchanged in these patients. Moreover, the differentiation or the maturation of these cells was reduced as the percentage of double cortin positive cells that expressed markers of differentiated neurons was reduced at the middle and late stages of the disease. And we thought that these results were very important and could have important consequences, not only for Alzheimer's disease, but also for other neurodegenerative diseases. So just to make a very long story short, I want to mention also the results we obtained on immature neurons in patients with other neurodegenerative diseases. We also use double cortin, but in this case, we analyzed not only the presence of cells positive for this marker, but also the morphology and the positioning of these cells. So as you can see here, in contrast to the decrease in the number of immature neurons that we observed in patients with Alzheimer's disease, patients with ALS, Huntington's disease, and Parkinson's disease exhibited an increased number of immature neurons, whereas patients with dementia with Lewy bodies and frontotemporal dementia showed no alterations in this number. Does this mean that adult neurogenesis is increased in these patients? Well, we have several data supporting that this is not the case at all, starting by the fact that proliferative cells and neural stem cells are not increased in the same direction that these cells. But what is even most important is that the differentiation of these cells, which means the acquisition of their proper morphology and positioning within the granule cell layer, is altered in patients with all these diseases. As you can see here, the lack of proper differentiation of immature neurons is a constant in all the neurodegenerative diseases we have studied. And this includes not only Alzheimer's disease, but all these diseases in which the hippocampus is probably not the main target of these disorders. Importantly, in all these diseases, in parallel to this altered differentiation of immature neurons, we found an increased number of pyknotic cells which were undergoing apoptosis. And these alterations go in parallel with a profound alteration in the functioning of the dentate gyrus neurogenic niche, which is characterized by astrogliosis, as you can see here, an increase in the thickness of the capillaries, which probably reflect neuroinflammation, and a remarkable reduction in the phagocytic index, which reflects the phagocytic capacity of microglial cells. So all these data led us to conclude that adult neurogenesis is differentially altered by distinct neurodegenerative diseases. In fact, we observed specific, let's say, adult hippocampal neurogenesis signatures in patients with distinct neurodegenerative diseases, although the increase in apoptotic cells is constant in all these diseases. And also the alteration in the dentate gyrus neurogenic niche homeostasis. but other specific stages of this process are differentially altered in these diseases. So just as a summary, our work detected the presence of neural stem cells, proliferative cells, and immature neurons in the human dentate gyrus until the 9th decade of life. We revealed the existence of a dentate gyrus neurogenic niche, which is very similar to that of rodents. And we could also conclude that distinct neurodegenerative diseases trigger the appearance of specific adult hippocampal neurogenesis signatures and impairments in the homeostasis of this dentate gyrus neurogenic niche. I would just like to conclude by introducing you to my research group, which is currently increasing. We are 2 more people in the lab. I would like to thank all our collaborators who made this work possible, and of course, our sponsors. So thank you very much for your attention, and I will be happy to answer your questions now.
Thank you, Maria. Very, very nice data. While we wait for the questions to come through the chat, I have a couple of questions. Do you have any hypothesis of why the mechanism in Alzheimer's disease seem to be different from other diseases?
Well, I think the first evident reason could be that in Alzheimer's disease, there is a direct targeting over the dentate gyrus, which is not the case in other neurodegenerative disorders. So I think one possibility is that this damage, not only in the niche, but also in the connections from the entorhinal cortex, could damage somehow the maturation of new neurons. And in the case of other neurodegenerative diseases, it could be that a first plastic attempt to increase the generation of new neurons is halted somehow by other molecular mechanisms that make that this increased addition of new neurons is not possible. So I would have expected that the number of cells detected in all these diseases would be different.
Thank you. I have another question from the chat. Is there any influence of the effect of age could be also linked to specific mutations that are associated to neurodegenerative diseases, to the ones that you look at?
Well, that's a very good question. I think that it has been shown that specific mutations in microglial cells, for example, are directly linked to changes in adult neurogenesis. And we think that this impairment in the phagocytic capacity of microglia to remove these dysfunctional or apoptotic cells is strongly related to the impairment in adult neurogenesis we observe not only during physiological aging, but also in these diseases, because this is something that we constantly observed in aged and diseased subjects. So it could be that one of those specific mutations that Michael mentioned before could be involved in these alterations in higher risk or higher susceptibility for impairments in neurogenesis during aging.
Oh, thank you. And last question, if you can have a few seconds to answer. There's a question about whether these changes are dynamic, whether they change over time.
Yeah, absolutely. I think they probably change over time, and also probably the alterations over the general process can change. It has been demonstrated that distinct stages of adult neurogenesis are regulated independently, both by physiological stimuli such as physical exercise and also by different type of insults. So it could be that over the course of time we found different alterations. In fact, that's the landscape we find in Alzheimer's disease patients at distinct stages of the disorder.
Thank you. Very interesting data and talk. Unfortunately, we have to move. So thank you, Maria. We have to move to the next Data Blitz session. The talk will be given by Kamen Tsvetanov, who is a research fellow at the University of Cambridge. And the title of the work is Resilience of Functional Connectomes in Presymptomatic Frontotemporal Dementia.
Hello, my name is Kamen Tsvetanov, and today I'll be talking about resilience of functional connectomes in presymptomatic frontotemporal dementia as part of my work as a brain non-clinical fellow at the Cambridge Center for Frontotemporal Dementia. I have no interest to declare. FTD with 30% of genetic cases allow us to study the consequences of disease processes before symptoms, while family members provide closely matched mutation-negative controls. Recently, we showed that functional integrity in 4 higher order networks is related to performance across a wide range of cognitive tests. And this relationship was stronger for presymptomatic carriers shown in orange relative to their non-carrier family members shown in blue. However, it remains unclear whether it is the integrity of these networks or it is the integrity across all brain networks that enable individuals at risk of dementia to sustain cognition despite brain-wide atrophy. And so here we were interested in ways to characterize brain-wide functional integrity and its relationship to performance. But brain function is a complex issue. It's a multivariate problem with many brain regions which are active over time and often summarized in functional connectomes. To further make sense of these connectomes, our recent work in healthy aging shows that diffusion mapping, which is a data reduction technique such as principal component analysis, could be a useful way to characterize brain-wide gradients of connectivity. Here, I'm showing 2 gradients which confirm that the activity between unimodal and transmodal regions is different from one another. Importantly, if we integrate all 3 gradients together, we can see that the brain regions in blue recapitulate the 4 large-scale networks in our previous study. This multidimensional gradient space allows us to explore the organization of functional communities such as the 7 large-scale networks defined by Lleó and colleagues. For example, using Euclidean distance to estimate similarity between each region in this multidimensional space, we can quantify the integrity within and between the Lleó networks. By doing that, we found a strong decrease in the integrity within the networks in symptomatic carriers compared to presymptomatic carriers and non-carriers. But there were no differences between presymptomatic carriers and non-carriers. This maintenance of network integrity in the presymptomatic carriers was behaviorally relevant and independent of the degree of brain-wide atrophy. In summary, we showed that the functional gradient framework provides a robust and standardized quantification of individual variation in brain-wide functional organization. We found that symptomatic frontotemporal dementia changes the integrity of network connectivity, while the maintenance of network integrity in the presence of presymptomatic neurodegeneration may be an important factor to protect against clinical conversion in individuals at risk of dementia. With this, I would like to thank you, and thanks to Guarantors of Brain, John Rohrer and the GenFi team, as well as James Rowe and the Cambridge Center for Frontotemporal Dementia.
Thank you, Kamen. Very nice data, very nice work. We have time for one short question. I have a question whether neuronal network integrity can be considered a biomarker. It seems that it's a good sensor for symptomatic phase of the disease. Do you foresee any applications in trials or in these subjects?
Yes, the gradient framework provides, I think, a really very good tool for identifying individual-based estimates of functional connectivity. But while I think the possibility of having biomarkers, I think the focus here for us is really identifying, understanding the mechanisms of the disease in the presymptomatic phase and also the disease progression.
Okay, thank you. And I think we have to move to the next session. So thank you. Very nice data, Kamen. Now we have to move to the last talk, which is the clinical translational research talk. And the presenter is Betty Tijms, who is an associate professor at the Alzheimer's Center, Amsterdam in the Netherlands. The title of the talk is Clinical Heterogeneity in Alzheimer's Disease, Implications for Clinical Trials. And we will have 5 minutes for questions at the end.
Hello,
Betty.
I'm Betty Tijms from the Alzheimer's Center Amsterdam, and I will talk about clinical heterogeneity in Alzheimer's disease and its implication for clinical trials. These are my disclosures. Alzheimer's disease is pathologically defined by the presence of amyloid plaques and tau tangles in the brain. The precise causes for this pathology remain largely unknown, but the genetic architecture is complex, and this implies that multiple biological processes might be disrupted. How to study this in patients in vivo is an important question to investigate. And cerebral spinal fluid proteomics might help. In the CSF you can measure amyloid and tau, and at normal levels are robust biomarkers for the disease. And just using amyloid p-tau and t-tau, the first studies have identified that at least 3 subtypes might exist, or even 5 when ubiquitin was also included. But many more proteins can be measured in the CSF. And so the question is, can you identify more subtypes and also understand a little bit better which biological processes might be involved? And so we pursued this question in a large sample of 228 individuals with Alzheimer's disease across the clinical spectrum. And in these people, their CSF, we measured with tandem mass spectrometry proteomics. 708 proteins were identified in all the cases, and 565 proteins were associated with Alzheimer's when comparing to controls. We use those proteins to cluster, and we use non-negative matrix factorization for this, and this is a dual clustering method. So it means that proteins and individuals are clustered at the same time. And these are the results, and I'm going to focus on this heat map in which every row reflects one protein and every column a subtype. The color indicates the average levels for a particular protein within a subtype, and these values are scaled to a control group so that red levels represent concentrations higher than controls and blue lower than controls. So here we see the proteins that were specifically increased in subtype 1. And it is of note that exactly those proteins were decreased in subtype 3. And these proteins were associated with neuronal plasticity related processes, including cell adhesion, axon development, and the regulation of the MAPK cascade, which is important for neuronal plasticity. The second subtype showed increased levels of proteins that are associated with the innate immune system, and this included complement activation. A third subtype showed increased levels of proteins that were associated with a dysfunctional blood-brain barrier. We then compared the subtypes on clinical characteristics and found that they all had a similar proportion of people with normal cognition, mild cognitive impairment, or dementia. So they do not reflect disease severity. Rather, these biological processes seem to be disrupted very early on in the disease and do not necessarily change with severity. The subtypes also show a similar proportion for the ApoE4 genotype, and this suggests that it didn't influence the distinct subtypes per se. What we did observe was that there were quite large differences in total tau and p-tau levels, with subtype 1 showing mostly abnormal and very high levels of tau, and subtype 2 and 3 more lower levels and more normal. Now, since with data-driven methods you can always find subtypes, it's really important to replicate results in independent data sets. And this is where we used ethny. There a little less proteins were available, but still we observed largely the same subtypes. And here you can see that these subtypes also showed very similar differences in their total tau and p-tau levels. And the nice thing in ethny was that they had base 1 activation data available. And what you can see here is that subtype 1 showed increased levels of base 1 and subtype 2 and 3 showed decreased levels. Direct implications, because you might know that base 1 is the secretase that leads to the amyloid beta 142 peptide, which aggregates into plaques. And the trials that inhibited base activation showed very negative effects. So the treatment groups, they declined faster than the placebo group. Now, it might be possible that possibly in those trials, maybe subtype 2 and 3 were overrepresented, who have lower base levels than controls. And I can imagine that further inhibiting base in these individuals might be detrimental. Whereas perhaps these inhibitors might still be used for subtype 1. And it would be really great if we could test this hypothesis in existing trial samples. But in any case, we can see these distinct biological processes being associated with every subtype, and there might be even more opportunities to tailor new drugs according to the processes that are disrupted in an individual patient. Now, I mentioned that the subtypes didn't differ in terms of disease severity. And this might suggest that they could reflect potentially differences in genetic vulnerability. And so if this is the case, you should be able to observe it in controls. And for this reason, we repeated subtyping in a group of older individuals with normal cognition and normal amyloid and tau markers. And in this group, we also observed 3 subtypes, with the first subtype being really similar to the Alzheimer's subtype 1, showing increased levels of proteins associated with neuronal hyperplasticity. And we had repeated amyloid and tau measures over time available for these individuals. And these subtype 1 individuals, they showed over time aggregation of amyloid and also increases in tau, which we also observed in Alzheimer's types. Now, there was a second subtype that was more difficult to characterize, and it did not clearly correspond to the innate immune activation subtype we found in Alzheimer's. Possibly this might mean that this subtype might be a response after amyloid has aggregated. It could also be that it was just not present in this sample. And then the third subtype shows indications of blood-brain barrier dysfunction, really similar to the Alzheimer's subtype 3. And when we looked at the repeated amyloid and tau over time, we saw aggregation of amyloid only and no changes in p-tau, which was also similar to the Alzheimer's subtype 3. Now this suggests that there might be different sets of genes involved, but this also kind of highlights that there are possibly new opportunities for primary prevention. Perhaps these pathways influence amyloid aggregation or p-tau, or maybe they reflect the process of very early pathological changes. And if we better understand this, we might be able to stop amyloid from aggregating and tau from rising. But we're not stopping there. So we've got new data in a new independent data set which is larger in terms of individuals included, but also in terms of the protein measured. So also here we measured with 10ms tag spectrometry proteins, but with a newer machine which enabled to identify the double number of proteins. And so we could also double the number of proteins that were associated with Alzheimer's disease and use that for clustering. And what we see now is 5 subtypes. 3 of these subtypes showed increased tau levels and 2 subtypes showed decreased or normal tau levels. So the first subtype is really similar to the first subtype that I presented in the previous slides, showing increased levels of proteins associated with neuronal hyperplasticity. A new subtype also shows increases of those proteins and in addition shows signs of immune activation and also autophagy inhibition. Another subtype shows indications of microglia dysfunction and disrupted proteostasis. And then second subtype showed indications of innate immune activation, very similar to our previous innate immune activation, but because we measured more proteins, we now also see signs of choroid plexus dysfunction in this subtype. And finally, subtype 3 also appeared in this data set with individuals showing indications of blood-brain barrier dysfunction. And in this data, we also measured base 1 activation levels. And we see very high levels in the first 3 subtypes only. These were the subtypes that also showed high tau levels. So it seems that this increase in amyloid metabolism is also interlinked with higher t-tau and p-tau levels in the CSF. Base 1 was lower in subtype 2 and 3, like we also previously observed. So this replicates our previous findings. And with that, I'd like to conclude. And I hope that you appreciate that robust CSF proteomic subtypes exist. We replicated these subtypes now in 3 independent cohorts with different multiple mass spec MS techniques. And so that means that the subtypes that we find are not a particularity of the measuring technique that we use, but I think that they really reflect robust biological subgroups. We also show that with increasing your sample size and increasing the amount of proteins measured, that's possible to detect new subtypes and further refine the old ones. And very recently, a tissue proteomics study using completely different techniques also found similar subtypes. That further supports the notion that with CSF proteomics we're really picking up this clinical heterogeneity, and we are able to disentangle this for subgroups. Our results have clear therapeutical implications. It makes sense to develop drugs that, you know, might help particular patients who show disruptions in specific biological processes. CSF proteomics might help not only in identifying these subgroups, but also perhaps for stratification to select those individuals who will most likely show a treatment response. Tau by itself won't do the trick, because we now see 3 subtypes with similar tau levels, but they have very distinct biological processes disrupted. it would be great if we could further test of whether this would really help with treatments. And the thing to do is to try and test these hypotheses in existing trial samples. So if you are aware or you have access to such samples, please contact me and perhaps we can collaborate. And with that, I'd like to thank all my collaborators and thank you for listening.
Thank you, Betty. Very nice talk, very nice work. We have time for a few questions. There is one in the chat about the association between these proteomic subtypes and the clinical subtypes of AD, let's say logopenic or posterior cortical atrophy. Is there any association between both?
Well, in the first paper where we found 3 subtypes, we couldn't see very clear differences in the subtypes in their clinical symptoms. Not to the extent that you usually see with atrophy patterns. So there you find subtypes too, and that's very clearly linked to the symptoms that people develop. So I'm not sure if that's because we simply do not pick up the proteins that might be specific to certain brain areas that can explain regional vulnerability. So in the new data where we have many more proteins, maybe there's a sign in there as well, but I haven't gotten there yet in the data analysis.
Thank you. I have another question about whether these changes, these subtypes, also are stable over time. So if you repeat the lumbar puncture, can they change, or is there any data whether they change or they keep in the same subtype?
Yeah, and that's a good question. One of the reviewers of the first paper also asked that, and so in ADNI, there was a really small subsample of people who had repeated proteomics available that also overlapped with the subtypes that we found. And for them, I made this composite score of the proteins that were specific to their subtype. And that remained largely stable over time. But I think, you know, there should be some changes at one point. As you progress through your disease, you simply lose brain cells. So that should impact on the protein concentrations. So yeah, largely an open question still.
Thank you. And I think we have time for last questions. We have some seconds left. The question is, are the subtypes mutually exclusive? So whether they have elements of neuroplasticity and immune response, or there's a lot of overlap between them?
Yeah, well, so we put a hard threshold in there, but in principle, every subtype can have a probability. Only that makes our life much more difficult if you have these continuous probabilities for several subtypes. But indeed, it's not the case that everyone is very clearly separated. So it would be very interesting to see, or also for trial development, you could think of just looking at the probability for a particular subtype for an individual. Yeah.
Okay, thank you. Thank you very much for the questions. So I think we have reached the end of the session. I would like to thank all the presenters and delegates for joining the conference, and I just want to remind you that you can visit the posters and you can take the opportunity to meet other delegates in the platforms and join roundtable discussions. So thank you very much for your attention.